Dear students, welcome to your online classes. We were in genetics and before quarantine we have read about mutation. Today we will discuss DNA repair from unit 2. Here we are going to touch these points: introduction, what is damage, how DNA is damaged, types of damage, then last one is your types of repair. Let's start from its word derivation. What do you mean by repair? We can see here that there are two words. One is DNA, another one is repair. We know about DNA, so here we have to know what is repair. Repair is the word. It means restore and put back in. As I have written here, repair meaning is restore and put back in. It is derived from Latin word re and parare, parer. First one is re, second one is parare, which means in Latin re means again and parare, parer means make ready or prepare. Then is there any repair needed? If there is no breakage or no damage, if there is no damage, there is no breakage, is there any repair needed? No. So where repair is needed? If there is any parts get damaged, then repair is needed. So it is your DNA structure. Just to understand, it is the rough structure. DNA is a molecule. As we all know, DNA is a molecule. This molecule contains nucleotides, and nucleotides made up of phosphate group, sugar group and nitrogenous base. Which are those nitrogenous base? ATCG: adenine, thymine, cytosine and guanine. So if there is any damage found in DNA, then all the processes, there are some process that conduct DNA repair mechanism to repair the damaged DNA and correct it again. Okay, so now we can understand, we have to understand that what is damage. Before repair, we have to understand what is damage. Damage is the word. It also derived from Latin word damnum, damnum, damnum. It means loss. So this word itself say that damage means loss. What are the causes of damage? Some environmental factors. Some environmental factors like radiations, physical stimuli, chemicals. These are the examples. Chemicals, then biological effect. Due to these causes, due to these reasons, the DNA get damaged. And due to this reason also, remember we already know in mutation that these are the mutagens also. Those reason cause mutations. Those are mutagens. These are those reasons only. The radiations, physical stimuli, chemicals, biological effect. Due to these reasons, the chromosomal aberration and genetic mutation is found. If genetic mutation and chemical chromosome aberration is found, then due to this reason only the DNA also get damaged. So here DNA damage on the basis of alteration involvement of nitrogenous bases. Here you have to understand nitrogenous base. What is nitrogenous base? I have written here. I have told to ATCG: adenine, thymine, cytosine and guanine. On the basis of alteration involvement, if any wrong nitrogenous base make any wrong pair, base pair, any wrong base pair, then it gonna wrong. It's gonna damage the DNA part. So if there is nitrogenous base involved alternately, means A always bind with T and C always bind with G. A always bind with T by double bond and C always bind with G by triple bond. This is the method, or this is the sequence. But if A is bind with G and T is bind with C, then is this correct? No, this is not correct. Due to this reason only the DNA get damaged. There are various reasons, but this is one of them. So here, on the basis of this, DNA damage is of two types. One is monoadapt and one is diadapt. Here see, mono means single, di means more than one. If one nitrogenous base alternate the DNA sequence, then it is called monoadapt. If one or more than one nitrogenous base alternate the DNA damage process or DNA sequence, then it is called diadapt type of DNA damage. So now we will see what the DNA repair is. The DNA repair is a collection of processes by which cell identifies and corrects damage to the DNA molecules that encode its genome. It is the definition. So we can learn like this, that DNA repair, the key points are collection of processes. DNA repair is the collection of processes that help the cell to identify and correct itself, correct the damaged part, and enable DNA to encode its genome. Those nitrogenous base ATCG, what is their role? They are forming the code, genetic code, and regulate the whole processes like replication, then translation, transcription. But if DNA get damaged, then is this possible further? No. So to repair the DNA, some enzymes are required, and some enzymes, with the help of enzyme, the DNA repair itself. So one of those is nuclease enzyme. Those nuclease enzyme is divided into exonuclease and endonuclease. Okay, you know already about exonuclease and endonuclease type of enzyme. So exonuclease is the enzyme, both the enzymes are help to catalyze the cleavage of phosphodiester bonds. Means ASE means it is the enzyme. We know if ASE is there, then it is the enzyme. It is the nuclease enzyme. It is divided into two, exonuclease and endonuclease. Both common process or common act is catalyze the cleavage of phosphodiester bonds. Means it break down the bonds. It requires the difference. Now we see the difference in between this and exonuclease and endonuclease, that this requires a terminus for hydrolysis. What is hydrolysis? Hydrolysis is a chemical breakdown of compound due to reaction with water. Is that clear? Due to after reacting with water, the compound is break down chemically. This is known as hydrolysis. And exonuclease, it requires hydrolysis. It requires a terminal point for hydrolysis. But endonuclease doesn't require. Cut off terminal nucleotides. It cut off the nucleotide which is in terminal. Terminal means in the end part. But here break internal bonds. It cut at last. If it is the DNA sequence, then it cut at last. But it cut in the middle. Which one? Endonuclease. Then endonuclease act on damaged DNA make it correct. Endonuclease, why these enzymes are present? These are present for help to DNA damage and make it correct. So this particular enzyme, endonuclease enzyme, act on damaged DNA. If the DNA is damaged in this part, if the DNA is damaged in this part, then the endonuclease enzyme make it correct. Okay, and the correction of the molecule is called correctional nucleases. This is the new thing. Correction of the molecule is called correctional nucleases. How it is cut and how it repair the DNA, that we will learn later.